Hi, and welcome to the final lecture in the Metabolism series. I kind of already said I would finish with the electron transport chain, and then I kind of went into metabolism, anaerobic respiration, and fermentation a little too quickly. So I thought I would follow it up with this video here, strictly on anaerobic respiration and fermentation. These two seem to highlight um, a lot of real-world processes here. And while every cell in your body aerobically respires in the mitochondria, it's important to learn anaerobic respiration and fermentation because they are pretty routinely seen in both the environment and in human processes. Okay, so what we're seeing with anaerobic respiration and fermentation is that they are two distinct entities. Okay, they are not the same. Anaerobic respiration still uses the electron transport chain. It uses glycolysis. It uses the Krebs cycle, and the transition reaction. It uses it all. The difference between anaerobic respiration and aerobic respiration is our final electron acceptor. We talked a lot about the electron transport chain and how most of our ATP is generated through oxidative phosphorylation. Well, you can't have oxidative phosphorylation if you don't have oxygen. So uh, that is the defining characteristic. Okay, so we will still have the electron transport chain, and we will still have the redox reactions, except our final electron acceptor will be carbon dioxide or nitrate or sulfate. Okay, that is the main difference between anaerobic respiration and aerobic respiration. Fermentation, on the other hand, is what happens when we can only get into glycolysis because we don't have oxygen available to us. Okay, without oxygen and without the machinery readily available to anaerobically respire, some cells can ferment. Okay, and fermenting is the action of getting back NAD to enter glycolysis. Okay, so it's like the recovery reaction. So when you say fermentation, you're talking about the reaction that happens after glycolysis, after we got our ATP from glycolysis. Okay, so these are the two methods that we use when we're not dealing with oxygen. So I kind of went into it a little bit in the last video and now, but we anaerobically have bacteria respiring in our guts. It happens in cows also. In their different stomachs, they have these containers just for anaerobic respiration, and usually that produces methane. Okay, and we can smell methane in the air. This is also in swamps, in bogs, and how you get that peat moss that stays there, all that nice organic matter that stays there can burn later. Okay, anaerobic respiration. These are places where there's no oxygen. Okay, so we get from glycolysis, we take our glucose, we go into glycolysis, we get to our transition reaction, we go into the Krebs cycle, and when we get into the electron transport chain, we don't have an oxygen there, and we dump our electron onto another electron acceptor, namely carbon dioxide or nitrate or sulfate. Okay, so this is common, and in lab I told you about how there are anaerobic respirators, these big large machines, well, containers rather, where they let natural processes occur where the oxygen is low and all the anaerobic bacteria are able to decompose our sewage waste, right? And it's a big old massive thing along the sewage line before they start to clean it. Then they clean it and then put it back into the ground. Okay. All right. But big anaerobic respirators, natural process, still just as much ATP as aerobic respiration, just a little bit less. Okay, just a little bit less. We're still talking in the 30s because we've entered the Krebs cycle, because we've entered um, the electron transport chain. Okay. Fermentation, on the other hand, is what you're thinking. It is not going past glycolysis. If you're an organism alive, you can enter glycolysis. You can take that glucose molecule and split it and get some energy from it. That is ubiquitous across all organisms, even archaea. All right, suggesting that in order to have become a cell, you needed to have been able to have that process. Okay. However, sometimes you don't have the electron transport chain to anaerobically respire. Say you're yeast, you're a eukaryote. You don't have anaerobic respiration available to you, but you don't want to die yet. Okay, so you start fermenting. You start another process after glycolysis to return to glycolysis again. Okay, if we need NAD in glycolysis, we need to get that back. How do we get that back? We get it back through fermentation. Okay, and that is when there is no alcohol, or there is no oxygen, but there is plenty of sugar available to them. So they would be happy otherwise, except for no oxygen. Okay, so this is the recovery reaction that you have. You have two different types. You can make lactic acid, and that is common in our muscles. Okay, we make lactic acid, and that is what gives us that muscle fatigue. All right, but what did our muscles do is we recovered our NAD. Okay, we wanted that NAD so we can get back to producing ATP through glycolysis. Okay, so now we have our NAD and we put it back all the way up into that top graph on the top left, back into glycolysis so we can ferment it again. Okay, and we want our NAD. Other times in yeast, we're going to produce ethanol. Okay, so lactobacillus and our muscles produce lactic acid, and that's in cheese and yogurt. Okay, and that's where you get they ferment the lactose into lactic acid, and the ethanol here is produced in yeast, and that's where we get bread and we get beer from. The bread, when you smell it cooking, is actually the alcohol burning off, right, and into the air, and that's what you're smelling. It's the alcohol burning off. Okay, so these are the recovery reactions, and we needed this NAD so we can go back and put this into our glycolysis reaction. Okay, and in my analogy, remember it's the full battery. We're using a full battery to get an empty battery. Okay, but this empty battery is more important because ATP is the better battery. Okay, so if we can't power things with NAD alone. Okay. All right, so fermentation versus respiration. Okay, and so when I say respiration, we mean anaerobic and aerobic. Okay, when we say fermentation, we mean just fermentation. Okay, so no electron transport chain. Okay, we can see that right here. Right, we don't go into the Krebs cycle or the electron transport chain, and we're all the way over here. Okay, so no electron transport chain, and we only get ATP through glycolysis or substrate level phosphorylation. Okay, in respiration, we use that electron transport chain that we talked about, the dam analogy. We pump protons and then we let them flow back down through our pump and our machine so we can turn a turbine. Okay, and that means we get most of our ATP through oxidative phosphorylation. Okay, so in glycolysis, which is during fermentation, right, in keywords, we're going to make 2 ATPs. Right, aerobic respiration makes about 36. Okay, anaerobic respiration makes about 32, 31 per molecule glucose. Okay, and the yield depends on the nature of the electron transport system and what you have going on environmentally. Sometimes, like fermentation, right, you don't have some substrate available to you, so you do an alternate process. So you wouldn't just make ATP if it wasn't as available to you or necessary to use in this specific process. Okay, and you can switch to that describes our Pasteur effect, because you can switch from fermentation to aerobic respiration depending on what you have. And there is a video I just watched to add to this point about fermentation, right? Historically, yeast make ethanol, right, in our beers and our ales in the abbeys and monks and monasteries in medieval times, right? And so you're dealing with yeast that can stand up to 2 to 3% alcohol. And once that alcohol gets too much, it actually starts to poison them. So they actually die and they won't produce more alcohol, right? And now we've bred yeast to withstand more and more alcohol, okay? All the way up to some yeast, like a specific strain of yeast, going up to like 16% alcohol. Okay, so that means they can tolerate alcohol as a poison and not die, and so they have ways of dealing with this poison. Okay, it's easy to say we bred something so strong, but nowadays we're dealing with like 6-7% beers, maybe even 8% beers in the craft beer style. So that's almost double or triple the historic yeast count. So yeast are very important in our bread and our beers and our society. And without yeast and fermentation, we wouldn't be here. And without anaerobic respiration in our sewage lines, we would be like the Romans and have to have maybe shared resources and a stick with a cloth on it, right? So we've come a long way by using these two processes. So I'm glad I kind of came back and revisited this topic. So again, here's our question. We're making beer, not producing alcohol. What's our most likely explanation? Our most likely explanation is... Alright, so the most likely explanation is that there's too much oxygen, okay? And so they didn't switch to fermentation, and so they are not making alcohol, because they would much rather have oxygen and enter the electron transport chain than just ferment. Alright, so hopefully this clarified fermentation and anaerobic respiration, and yeah, I'll see you next in nutrition.